Hello and welcome back to my channel. In this part of the video, we are going to discuss about Mendel's law of inheritance. In the previous video, we have discussed about what is genetic, what are the basic terms that are used in genetics, what do you mean by genetics, who was the father of genetics, and certain important terms I have discussed in the previous video. So I request you all to first watch the first part of the video, and then you can easily understand this term. So let's begin with the Mendel law of inheritance. Before that, let me tell you that Gerard John Mendel was the father of genetics, who gave the term genetics. He postulated certain experiment on the Pisum sativum, that is the pea plant. Okay, he conducted an experiment. By conducting experiment, he made certain laws which is known as Mendel law of inheritance. What are this law and what is he concluded? We will be dealing with it now. So let's begin. First law is law of segregation. Before knowing that what is this law, says that if you understand in the simple English meaning of the word segregation, means to separate. Okay, so simple meaning of the word segregation is to separate. Okay, so simple meaning of the word segregation is separation. So he made a cross on the monohybrid cross. What do you mean by monohybrid cross? A cross which is made by using a single trait. Let's say, for example, that is the height of the plant. If we make a cross by the using the height of the plant, that is capital T, in cross, that is small t, that is we are using only a single trait, that is or character, that is the height of the plant. That type of cross is known as the monohybrid cross. This is a very important point to be noted down. He saw that each individual possesses two character. You can see here, this is the mother, suppose this is the father, male, this is female. You can see here that is both of them possess two characters. Okay, two factors, capital T, two are present, but during the formation of gamete, while the gamete are formed, each of them will contribute only one factor, that is one gene will be contributed. So the progeny which will be formed will have the characteristic one from the mother, one from the father. This is known as the segregation. So while the gamete formation, the individual will segregate. This is only written in the statement. States that each individual possesses two factor, that is gene, that is you can see here two factors are present, capital T, small t. At the time of gamete, each membrane of the pair of genes separate from each other, so that each gamete carry only one factor or gene. You can see here the gamete which is formed carries one factor from the father, that is capital T, and one factor, that is small t, from the mother. This is known as the law of segregation. Okay, so first what he did in the P generation, that is in the parent generation, he made a cross between the tall, that is male, and a dwarf female. So the gamete which was formed from this tall, that is a single T was formed. Okay, from 2T was present in parent generation, but when the gamete was formed, only a single T was formed. This is known as segregation, that is the only single factor will be passed on to the gamete, so that the progeny will be formed is carries one factor from father and one from the mother. You can see here also, that is small t, only one factor is transferred, that is small t. When we make a cross between this capital T and small t, we get this tall plant, because we know that capital T, that is tall, is dominant over the dwarf. Then this is known as the F1 generation. Okay, wait. Next, when he made a cross between the in the F1 generation, he made a cross again by this hybrid cross only, that is capital T small t and capital T small t. He found that the gamete again from capital T one factor and small t one factor, and here also the same. Okay. Next, he made a cross, that is between the capital T small t. This is known as Punnett square to know the ratio at the probability how many dwarf or how many tall plant will be formed. So he found that 3:1 ratio, that is phenotypic ratio, was 3:1, whereas the genotypic ratio was 1:2:1, that is 1 tall and 2 was hybrid one, okay, and 1 was the dwarf one. So 1:2:1, and if you, in short, if you say 3:1 means the genotype is 3, that is 3 tall and 1 dwarf. Okay, so this is known as the law of segregation, that is during the each gamete, each male factor carries two factors, each gene carries two factors, but during the gamete formation they separate out. So the ratio which we obtained is 3:1. Remember the ratio, which is very important. Okay, next thing is here which was discovered was the test cross. What is a test cross? A cross which is made between the individual of the dominant phenotype and the recessive phenotype. Okay, so one dominant and one recessive phenotype, the cross which will be obtained is known as the test cross. Okay, so test cross is between the individual with the dominant phenotype and the individual with recessive phenotype to see if the individual with the dominant phenotype is homozygous or heterozygous. That is to find out whether the formation is heterozygous or progeny is heterozygous or homozygous. For that, this process is done. For example, if you see that if you take first example is homozygous, that is capital T small t, this is recessive one. Okay, so the first progeny which was formed in the F1 generation was tall, that is heterozygous condition was formed, which was dominant. Second, when he took heterozygous condition, that is capital T small t, and dwarf one, that is small t, because test cross means always it will be crossed with the recessive one. Okay, but first case he took the homozygous condition, that is capital T. Second, he took the capital T and small t, that is heterozygous condition. He found that one was tall, one was dwarf, that is he found one tall plant and one dwarf plant. So by this he concluded that the tall plant must be producing two kinds of gamete and therefore it is heterozygous. Okay, so by this he concluded the experiment. Okay, this is the first law. Second law of independent assortment state that by in this cross he made independently he crossed. Okay, this law states that in this he used dihybrid cross. Okay, dihybrid means two independent character, for let's say one for the color, another one for the shape of the seed. Okay, one color of the seed, one shape of the seed, these are the two different traits. So he took the two different traits and made a dihybrid cross. One gene occur independently of the other non-allelic gene at the time of gametosynthesis because new combination of character are produced. The simple meaning is that these two characters, that is the seed color and the seed shape, does not depend upon each other. This is known as law of independent assortment, that is they assort or separate out, they do not have any involvement upon one another, they separate, they have individual their role. Okay, they do not depend upon each other, that is the seed color and the seed shape do not depend upon each other. So he made a cross. First cross he took yellow round seed, yellow was defined the color, round seed means shape of the seed. Second, he took green color and wrinkled seed. Green defines the color of the seed, wrinkle defines the shape of the seed. This is known as dihybrid cross because they took two different character. Then when he made a cross, he found yellow red seed. Okay, yellow round seed because yellow is dominant over green and round is dominant over wrinkled. As I have shown you in the previous video, that chart I have made, that one you remember, this will be very clear. Okay, then what happened in the F1 generation, this gametes was formed. Then when the F2 generation, he got the ratio, that is 9:3:3:1. Okay, he got this ratio by this doing a Punnett square in the F2 generation. Then he made a dihybrid test cross the same way as that of the test cross, that is the round and yellow and wrinkled and green because this was the recessive one, and he got the following result, that is 1 is to 1 is to 1 is to 1. Okay, that was the result which he got from the dihybrid test cross. Then what do you mean by incomplete dominance and codominance? Incomplete dominance, in which if the two allele, that is both recessive and the dominant allele, is not expressive, that is no allele is dominant over the other, is known as the incomplete dominance. What is incomplete? A new variety will be formed. Okay, that is known as incomplete dominance. That is both the allele will not be expressed and totally a new variety will be formed. That is why 1 is to 2 is to 1. Example is Mirabilis jalapa, a plant in which red male flower was taken and white female was taken and a cross was made between them. But the F1 progeny which was found was pink in color. Why it happened so? Because of the incomplete dominance. Because neither the male red color male nor the female expressed each other to dominate each other. They did not dominate each other. That's why a totally new variety of plant was formed, that is pink color plant. Okay, and when he made a cross between them, he found in the F2 generation a ratio that is 1 is to 2 is to 1 ratio. Then what is codominance? That is if the allele lack the dominant and recessive character and both are equally shown or expressed, that is both dominant as well as recessive, both are equally expressed, is known as codominance. Example is in the case of MN blood group, which is seen, these are the codominant, that is both of them will be expressed at a time. Both the allele will be expressed at a time. Incomplete dominance means no one will be showing dominance, that's why new variety will be formed, whereas codominance means both will be dominant. So this is all about certain laws of Mendel, and in the next part of the video we will be coming with the certain new topic regarding this genetic, and hope you are clear with this laws. And if you have any doubt, you can ask me in the comment section, share among your friends, and do like and subscribe to my channel. Thank you and have a nice day.